Oh, perfect. So today it's my pleasure to have Dr. Samuel Revolinski with us here in one more day at the International Weed Genomics Consortium webinar. So Samuel is an assistant professor of weed physiology, biochemistry, and molecular genetics at the University of Kentucky. He did his bachelor degree in plant science, University of Minnesota, and then he completed his PhD in crop science at Washington State University. His doctoral thesis explored the genetic controls of flowering time and theoretical models explaining the maintenance of genetic variation in the self-fertilizing weed downy brome. Dr. Revolinski continued his research as a postdoctoral researcher, working on genomic resequencing, genomics for population genetics in weeds, and a massive herbicide resistance screening project that screened bromes, tectorum, downy brome from across the state of Washington for resistance to various herbicides to map genes contributing to herbicide resistance in downy brome. At the University of Kentucky, Dr. Revolinski has developed a research program that explores mechanisms of herbicide resistance tolerance in crops and weeds, developing rapid genetic markers for diagnostics and adaptive genetic variation in weeds. So just as a reminder, this is the second to last webinar that we have for this webinar series. We have another last one for the year in November, and we'll for sure continue again next year. So it's just a break in December. If you have questions, please use the Q&A tool here at the bottom of your screen, and then I'm going to read the questions later for Samuel. Cool. Samuel, take it away. Thanks so much again for agreeing to speak to us, and looking forward to seeing your presentation.
Well, thanks for having me, Kyle. And it's my pleasure to present here at the International Weed Genome Consortium with this webinar. And so when Kyle asked me to present, I really started thinking about what would be useful to the International Weed Genome Consortium. And before I completed my PhD with Ian Burke at Washington State University, I worked for a statistical geneticist at Washington State University named Jiwoo Zhang for 2 years. And he's one of the he's the last author in the original GAPIT paper, which released a bunch of GWAS models. And he used to postdoc with Ed Buckler, where he learned about GWAS in plants. And so I thought I've really been trained in both GWAS and weeds. And I think understanding why and when and how to use GWAS studies will be a good opportunity for other weed scientists, especially those working in genomics. And yeah, big thing that would happen with the human genome when that was released is people immediately started doing genome-wide association studies, which I'll explain more about in a moment. But that's kind of why I thought I would give this presentation here today, and thank you all for being here to watch it. And so what I'm going to go over today is part one, what is GWAS? I'll give a brief history of GWAS, and then I'll show the evolution of GWAS models, where I'll show you basically how it started and how it continued. And then part two, we'll be talking about using GWAS as a technique for weed science. And so when is GWAS useful for weed science? And then what are the benefits and pitfalls of using GWAS in weed science? And then I'll give 2 examples from Bromus tectorum that I've been a part of. And then I'll give a short conclusion on using GWAS for weed science. And so to give a brief history of GWAS, people hadn't done any GWAS or genome-wide association studies before the first draft of the human genome in plants and animals, there was QTL mapping, or they called linkage mapping, right? And that was because you could create linkage maps based on controlled crossing, but that wasn't really possible in humans, right, for obvious reasons. And so in 2001, the first human genome sequence was published by the Human Genome Project and Celera. And so in the bottom corner of the page, I have a picture of Craig Venter. He's the founder of Celera. It's a genomics company, but he really had a big part in spearheading that effort. And then in 2002, a Japanese group identified 94 cases and 68 controls of Japanese individuals with myocardial infractions, and that's a heart disease or not. And so they used a simple chi-squared test of association, but that was the first true GWAS. And then the International HapMap project was completed in 2003, creating a database of 100 million genetic variants, or like SNPs, within and between different genetic groups of humans, right? And so that allowed for bigger GWAS studies across different populations to be performed. And so after that HapMap project came out, the GWAS um, manuscripts really started to increase in frequency, especially in humans, because it opened up this whole world of being able to actually map where diseases were controlled in the genome. And so they could get an idea of what genes controlled it and try to make personalized medical treatments based on what genes people have or what variants they have. And so to go on about the history of GWAS, we get into kind of where people started to do it in plants, right? So with humans, it really took off quickly because you could never do anything like this before, simply because you can't make controlled crosses of humans ethically, right? And so uh, a really groundbreaking paper came out in 2009 where Ed Buckler used a nested association mapping panel and did a GWAS with it to reveal the genetic architecture of flowering time in maize. And that was really a big deal because um, people hadn't really done that before in plants, and it really showed kind of this cool distribution of the allelic effects. And in 2011, a group of people, Edward Buckler was also part of this, developed what people call genotyping by sequencing. And so that's kind of RADseq methods to develop a lot of markers from Illumina sequencing, right? So you could get thousands of markers for hundreds of individual plants or genotypes. And so results from GWAS uh, have been used for plant breeding. And so one example of that is out of that NAM population that Edward Buckler made for corn, they found uh, variants that increased carotenoids. And so they made vitamin A rich corn using those results. But now a number of weed reference genomes are now available, so we can really start to think about these traits in a quantitative way in weeds. And so we'll start talking about the evolution of GWAS models, right? And so the first GWAS study that came out of Tanaka's group, they actually just looked at the changes, right? So they looked at the controls, right, versus the people who had the mitochondrial infraction and looked at the ratio of different variants, and then they could come up with a chi-square value, right, and get a p-value for that. It was a pretty simple test and didn't really correct for anything, but it was how the first GWAS test was done. And one thing to think about with early GWAS tests, especially in humans, is originally they're set up for binary traits. So either you have the disease or you don't. But when people move to continuous traits, they the test of association didn't really work out anymore, so they needed a new type of test. And so for traits like height or some of those early ones, what people started doing was a t-test of the correlation between the allelic dose, right? So whether, and the allelic dose is kind of abstract in that you're thinking about differences from the reference genome, right? And that's, you either have none, right? You have both alleles are the same. You have one allele is different, or two. It doesn't really matter which letter is where, but you're just thinking about the dose of a variant, right? And so you can actually calculate the correlation between that. And if you think about a correlation, it's really the um, covariance of those two variables over the standard deviations, right? And then so you get a R value, and then you can do a T statistic for the correlation coefficient, and you can use that T to get a P value. And that's one way early GWAS studies were done. Um, but one thing to think about that is you're doing this for every single gene that you have a SNP for. So if you have thousands of SNPs, right, you start to get into this type 1 error problem where you're getting into this multiple testing thing that causes type 1 error, right? So people started using corrections for the p-value thresholds. And so the simplest one is called the Bonferroni correction. And that's just you take your significance threshold, let's say 0.05, right? 0.05. And if you're testing 1,000 markers, right, then you just divide the 0.05 divided by 1,000, right? And if you're doing 100,000 markers, you divide it by 100,000. And that corrects for family-wise error rates. And family-wise error rates are just the probability that of all those independent tests you do, at least one is wrong, right? And so if you're doing a bunch of tests and each one of them has a 5% chance of being wrong, right, you're going to get a lot of them are wrong the more tests you do, or you get that family-wise error rate will rise. But the other side of the coin to this is that as you implement stronger and stronger correction thresholds, your power goes down. And so with this Bonferroni correction that's typically used in GWAS, um, it controls the type 1 error really well, but it can cause a big loss of power, especially when you're looking at hundreds of thousands of SNPs or millions. And so one thing to think about when you're interpreting GWAS results is that if you have um, significant results according to the Bonferroni correction, that's really trying to err on the side of preventing false positives, right? But by sacrificing power. So that would be like if a judge in a criminal court required a massive amount of evidence to convict because they would rather have some criminal slip through the cracks than convict somebody that's not actually guilty. And so going on from there, people started using what's called general linear models instead of just a t-test of the correlation. It became a t-test of the regression coefficient. And so if you think about one of these models, you have your response equals the actual value of the allelic dose times the coefficient plus the matrix of principal components, and then the vector of fixed effects for those principal components. And so they made these linear models so they could add principal components, right? And so thinking about those maze populations, uh, I found a graph of the first two principal components of a large maze population. And you can see that different populations separate out differently. And so when you're thinking about principal components, it's basically just taking like this main component over tons of SNPs, and you get these orthogonal components, right? That basically explain kind of the broader population structure, right? So then you can filter out or correct for those effects by having that matrix of the however many principal components you choose. Usually it's a few, and it's kind of ambiguous. There isn't a perfect way to choose the number of principal components to include in a GWAS model, but people generally choose a few. You're not doing as many as you can. And so then it goes through each marker and puts it in as that X test and then calculates the coefficient or the B test, right? And then the principal components stay the same the whole time, but it goes through and tests each marker, correcting for population structure. So that's how it's kind of a step up from just the t-test of the correlation. But then you can go one step further and you can do mixed linear models, which were introduced that so the genetic relationships between individuals could be controlled for. And so this idea really came from animal breeding, right? So they always knew the pedigree of the animals they were breeding. And so they could actually make these mixed effect models to kind of parse that out. And so you had this Z and U, right? And the Z is an incidence matrix of each individual genotype. So it'll just have a 1 for that genotype in a column and then 0 for the rest, right? For each row as an individual. And then the vector of unknown random polygenic effects. And so that's actually has a covariance matrix of G, which is 2 times the kinship times the additive genetic variation. But basically just think of a random effect that's accounting for how related each individual in the population is, right? So that's one step past the GLM. And these MLMs have been really popular models for GWAS because they take into account both the population structure and the kind of lower level relationships between individuals in that population. So usually when people think of kinship, they think this individual is the grandfather of this individual, these are brothers, right? And so on, right? And that's like a pedigree relationship, but these are made with the markers, right? So it's a little bit more exact. And so finally, we end with modern GWAS models. And so really one thing they discovered about MLM is having a relationship matrix with the the kinship of each individual compared to each other is really expensive for the power, and you lose a lot of power, although it does reduce the type 1 error, but you can actually compress those kinship matrix so each individual is in a group and you're looking at how those groups are related to each other, and that's called compressed kinship. And that can increase your power without um, increasing the type 1 error. But then the big thing that happened was incorporating multiple SNPs simultaneously when performing the test. If you remember a few slides ago, or one slide ago, that X test XB, right? That's only testing one SNP at a time. The modern models actually will go through kind of a stepwise regression algorithm to figure out um, which of those SNPs to include and then include them while doing the actual test, which is really nice because more often than not, traits are controlled by more than one variant. And if you don't include them in the model while testing each other, you get inaccurate results sometimes. And then finally, they were able to incorporate multiple SNPs and have compressed or modified modeling algorithms for kinship to improve power and computational performance. So um, B-Link and Farm-CPU are both modern algorithms in the GWAS package GAPIT. And so Ziwu Zhang here, I used to work for him, and he maintains the GAPIT packages and came up with B-Link and Farm-CPU. And how both of those models work is that they basically uh, have reduced kinship models, and B-Link doesn't even have a random effect model, right? It just uses all the SNPs it finds related to the trait and comes up with kind of a relationship type model. It's a little bit different. And then Farm CPU has a random effect, but it's only made with a smaller subset of SNPs. And basically, both of these don't overcorrect for kinship, but they also are much faster because they don't require kinship to be computed from all of the markers in the genome. So if you're doing modern GWAS studies, I'd recommend modern methods like B-Link or Farm CPU. However, using older methods like MLM, sometimes reviewers can like that because you'll get the kind of classic Manhattan plot look versus the modern methods filter out markers that are in close LD and push their significance down. And so there's advantages to both. Also, the um, methods like MLM, they have really small false positive rates, right? So you have lower power with those models, but you can have a lot of trust in what they find. And so going on from that, we'll talk about when GWAS is a useful method for weed science. And so the first thing that comes to mind is when making controlled crosses of a weed is not feasible. So working in cheatgrass throughout my PhD, it's an obligate selfing species, and it's nearly impossible to cross. I When I first started working for Ian, I got to access some data from a suspected cross they had made, and some postdoc had spent a long time trying to get across. And when we finally got the genomic data, we looked at it and it was all the same as the female or the mother plant. So then it was pretty obvious that it wasn't a true cross. So in a situation like that, if you can't make crosses, you can't really do QTL mapping or LD mapping, right? So GWAS might be your only option. The other thing to consider is you want to use species where the genotypes can be stabilized via seed production or asexual propagation, such as rhizomes, right? So you want to be able to repeat those same genotypes. If you spend a bunch of money to get a genotype for an individual in a population, if you can get seeds that are the result of selfing or rhizomes, right, you can basically maintain that population indefinitely and keep using the same genotypes for multiple studies. Um, the other thing is to think about is if you're going to have enough sequence genotypes to have enough power to detect loci for a given trait, given the heritability of that trait, right? So you want to make sure you have enough individuals. You're definitely going to be over 100, and even things in the kind of 100 range, like what I'm going to show you later, they require really high heritabilities to have enough power to map those markers or the marker associations. And then finally, you have to have genetic variation between collected genotypes of the weed, right? So if you collect a population of weeds and you sequence them, and there's no genetic variation and there's no phenotypic variation, you're not going to be able to map anything. So that's another thing to think about. And you can do GWAS for cross-fertilizing species. The only thing I would say is you can also do QTL mapping or linkage mapping pretty easily in those species, and you don't run into the problems with population structure or kinship in LD mapping or QTL mapping as much. So it might be smart to use those methods in those cases. And also in outcrossing species, it can be harder to stabilize those genotypes. So you might spend a bunch of money sequencing a population to do one GWAS, right? And then you can't really do anything more with it because if you keep growing those populations, right, they come from heterogeneous seeds, so they're not going to be the same genotypes every time. And so what are the benefits of using GWAS for weed science? Wild populations can be used to form association panels for GWAS studies. GWAS can be used in populations where it's not possible to perform QTL mapping. Measures of the effect of variants, which is something that RNA-seq cannot do. So you can't do RNA-seq and see how big of an effect an allele has in a population. And then it allows scientists to identify candidate genes and understand the genetic architecture for adaptive traits in weeds such as flowering time and non-target herbicide resistance. So what are the pitfalls? Well, weeds are often inbred and have gone through population bottlenecks, leading to large disequilibrium blocks that often include multiple candidate genes. So then you have to validate the genes later. It can be difficult to grow weeds consistently due to factors such as seed dormancy. No cheap S&P microarrays exist for weed species, so genotype costs are higher than if you're working in something like soybean or wheat. And then some of the weeds are obligate outcrossing species, thus replicating a single genotype multiple times may not be possible. And so I'll give a couple of examples from studies I've worked on. And this is actually from the study where we released the reference genome for downy brome with Craig Coleman and Jeff Maugan at Brigham Young University, and Ian Burke was also on this paper. And so the first thing we found when we resequenced all this downy brome is there actually was quite a bit of variation between regions and populations. And you could see the principal components basically splits apart the Grand accessions, the Montana accessions, and the Washington accessions. And then when we did DAPC clustering, we saw that there are a lot of specific genetic clusters in this population. And then when we actually looked at the average flowering times for each of these populations, you could see that between Washington, Montana, and Grin, there is differences. And specifically, the Montana populations took longer to flower on average and were also shorter on average. And so basically we grew them in the greenhouse and we just waited to measure their height at the end and then also the number of days till it reached all these different phenology times. So VPN is the days till first visible panicle. J1 is the number of days till the first joint appeared. FRS is the number of days till the first ripe seed appeared, and then on 50 is the number of days until 50% of the seeds on a plant were ripe. And so we basically plugged them into this linear model. It was actually a mixed model where pretty much everything except for the genotype was a random effect, and then the genotype was fit as a fixed effect, so we could get the blues to use in R. And so this was all replicated. Uh, there was 3 reps or blocks within each greenhouse, and then there was 2 greenhouses or this environment. There were 2 trials to this, right? And so when we actually did these marker associations with B-Link, we could find a bunch of hits, and specifically for all of the flowering time traits or phenology traits, we found this hit on chromosome 6, right? And so we actually mapped out the LD over the distance in the chromosomes, and we figured out that we should probably be looking from about 250 kb to 500 kb. And so we figured out all the candidate genes within those windows, and that HDR1, that's the one on chromosome 6 that appeared in all of those traits, and it's actually the candidate gene is an ortholog of a heading date repressor 1 in rice. And so in 3 of the traits, it was 212 kb, and one of them, it was 12 kb away. And then other than that, we found gibberellic acid biosynthesis genes, which also control flowering time in barley, which is closely related phenologic or um, phylogenetically to downy brome. And then we found all of these FRS and FHY genes, which encode a transposable, a transposon-related gene that also regulates expression of far red light. Some other weed scientists, I think Eric Patterson found this group of genes to be related to herbicide resistance, which is pretty interesting to see. And then the second example, this is from the same population, but we just repeated and sprayed it all with sulfosulfuron, was looking at sulfosulfuron resistance in this population, right? And so the first thing we did was think about from the year 2000, what group 2 herbicides have people been applying to winter wheat in Washington? And so we dug this up out of the National Agricultural Statistical Survey, or NASS, for Washington State. And we were trying to figure out why sulfosulfuron started out so popular and then went down in popularity and was replaced by things like imazamox. And one thing we found that we didn't expect here is that people have been using a lot of mesosulfuron methyl. And so that was interesting to see, but we're wondering why so quickly did the sulfosulfuron get phased out. And so we decided to do a GWAS for sulfosulfuron resistance in downy brome. And so we had 2 trials in separate greenhouses like we did for flowering time, right? And each of those had 3 blocks or 3 reps within it. And we also had 3 doses and a control for every trial. And then the phenotype we actually measured was regrowth as a percent of untreated control assessed after 2 weeks after cutting, right? So we cut 21 days after treatment and then saw how much they regrew. And we also cut the control, so then we could compare the amount of biomass that the control group racked after the 2 weeks after being cut compared to the treated plants. And so we actually planted this with this high throughput planting using a shop vac planting boom, right? So it's basically, it accumulates a negative pressure with a vacuum and it has these modified pipette tips. And we put out the randomized flats in the order we wanted. And then we could just dump these seeds into new flats in the order we want using this boom. And then we have the tinfoil over them so we could do one row at a time and not drop the seeds into other buckets because then we'd be putting the wrong seeds in places. And so when we actually got these traits back, we could look at the distribution and correlation of the regrowth percent compared to the untreated. And what we found that was interesting phenotypically is that at these 3 doses, right, even at the low dose, most of the plants did pretty bad. This is 1/10 of the label rate. Most of them really didn't regrow, but there is this group of them that in all 3 rates survived pretty well. And you can see that there's this elbow shape to these plots because the trait is unrelated early on, right? But then as you get to higher levels of the regrowth, it's highly correlated between the 3 doses. And so when we actually did GWAS, and this is finally the Manhattan plot, right? So it shows all the chromosomes we have, and then across the chromosome, within each chromosome, it's the position in the chromosome. And then on the y-axis, you have the negative log 10 of the p-value. So the higher those values are, the more significant it is. And then we have these QQ plots, which basically is the distribution of p-values compared to the negative log 10 distribution of p-values, the observed versus the expected. And what it should be like a regular QQ plot where it should follow that line the whole time, but at the end, your significant markers should pop up out of that basically line. And so these are some of the best QQ plots I've ever seen. It follows the line perfectly until the very end where it pops way out. And that's exactly what you want to see in GWAS. And so this is pretty untypical in how significant this marker on chromosome 7 was. And we found it at all 3 doses, which is a good sign. And so it's nice we identified the same SNP in all 3 doses. There were a number of other SNPs, but one thing that was pretty incredible was that that SNP on chromosome 7 explains up to 48% of the phenotypic variation. And it's a semi-rare allele, right? So it's only present 9% of the time. And then, so this is generally how GWAS results are presented initially, right? You have a table with your trait, the method, the chromosome, the position. The Q is the corrected p-value. It's a different method than the Bonferroni. It's called FDR corrected or false discovery rate corrected. MAF is the frequency of that marker or the minor allele, right? So uh, an NMAF of 0.1 means that uh, that allele shows up about 10% of the time. The effect is actually just the raw effect, right? So that has to do with the trait itself, and then the PVE is the amount of the phenotypic variation that's explained by the marker. And so generally in GWAS studies, anything above 20% is pretty high for PVE. So having up to 48% in this study is actually pretty high. And so when we look at the candidate genes underlying these marker trait associations, there is one really obvious one, and that's a PP2A1 that was about 40 kb from that SNP that kept appearing on chromosome 7. And so the ortholog for that is actually that we found in the genome nearby is the serine threonine protein phosphatase, right? And so a phosphatase is basically, or that specific phosphatase regulates stress responses through phosphorylation, which means pulling phosphate groups off of protein substrates. But we also found several glycosyl transferases, a couple of cytochrome P450s, and an ABC transporter. And those are all pretty well-known gene families for non-target site resistance. And so kind of what we're concluding is that large effect stress regulation genes may be underlying sulfosulfurone resistance and bromus tectorum, although typical non-target site resistance mechanisms are also likely present. And so kind of the conclusions of all of this is that modern sequencing methods and improvements to GWAS models have made GWAS increasingly more feasible for weed science. GWAS is an excellent tool to examine the underpinnings of adaptive traits in weeds, although molecular validation of genes is required. If you find candidate genes, it would be really cool if people started testing these, and that's one direction I really want to go in my program. And then GWAS works well in self-fertilizing weeds that have genetic and phenotypic variation, right? So that's what I showed with those two bromus tectorum GWASs. And those are pretty small populations for GWAS. So unless those were large effect markers, we wouldn't have found anything. And then non-target site herbicide resistance may include stress response mechanisms in addition to metabolism. And so that's all I've got for my presentation.
Excellent, Samuel. Thanks so much for showing us such a comprehensive set of slides and your research. It's much appreciated. We do have a few questions here in the Q&A box, and I encourage the other attendees to also submit their questions. I want to break the ice, and can I ask you the first question? I do have a question about your LD slide, and because sequencing is too expensive, especially when we are talking about for GWAS, you want to have enough coverage, plus those are big genome species like Downy Brum is what, more than 2 gigabases.
I think it's
so can you
3.
3, so it's crazy, right, compared to other species. So can you use the information you have on LDDK to better inform you to do more like RedSeq GBS, more restriction or like a sampling of the genome, and that will help you to decide how dense your SNP data set has to be, or you still think we need to resequence the whole thing?
Well, I think what knowing the LD could do, or the LD decay could do, would be help you decide on what type of genotyping method to use and the depth to do your sequencing to. And because if you have large LD blocks, really what you're mapping is those LD blocks in association with a trait. So if you have large LD blocks, you don't need to do as thorough of sequencing, and you don't need as high of a density of markers. However, then you have a bigger candidate gene window to look through. But I think that if you have big LD blocks, it could allow you to use less deep RADseq type data versus really dense whole genome sequencing, like shotgun approaches.
Yeah, that makes sense. And then maybe instead of saving money, maybe you can include more populations or more genotypes.
Yeah.
That could be an approach. I'm going to read the next question here. A compliment. That's a great webinar. Is there a way to use GWAS approaches for new case of herbicide resistance where there may be only a handful of populations identified?
So it really depends on if those handful of populations are resistance coming from kind of the same mutation or the same haplotype block, right? So if you have a handful of populations, right, and the resistance in those populations arose independently, then the minor allele frequency of that variant might be too small to actually do a GWAS for. And in that case, I would recommend using something more like QTL mapping, right? Because then you can take that rare allele that's in that one population and do a cross to a susceptible population. And then the offspring from that cross, you should end up with like a 50% minor allele frequency for the variant that causes herbicide resistance. It's hard, you'd have to have really big populations of those limited populations to do GWAS with if they independently arise. If you have enough populations where it's the same origin of resistance, then you can do it. The other thing to think about is, so I've been doing some simulations with minor allele frequency and the power to find markers and GWAS, right? And you can take basically your association panel and you can simulate markers on top of them and then try to find them and see how much power you have. One thing I found through some of the simulations I've been doing recently, and this is for um, the sulfosulfuron resistance GWAS, we submitted to a journal and they want some more edits on it. And one of them was about power, but we could actually get decent power, about 50% for uh, a rare allele that occurred 10% of the time in a population in Downy Brome or in our association mapping panel. We get about 50% power, right? So with bigger populations, you'd be able to get more, but you should also think about simulating these on your own populations so that you can see kind of what numbers you need to get to as far as minor allele frequency and all that.
So I think that your answer is correlated with the next question, which is what's the total sample size you would suggest for GWAS to give us high analysis power?
So the best answer I can give is as big of a population as you can get. Um, the one thing that's pretty nice about GWAS is they do so much of this correction for the false positive rate that even in small population sizes, so if you're in like 100 or 200, right? The issue you'll run into is not finding markers versus finding false markers. And I saw a really good presentation at PEG on this a few years ago. One of Jiwoo Zhang's friends, when I still worked in his lab, has this whole started making presentations about how you can trust associations found in small panels, but if you set up experiments and you use small panels, you might not find any markers, but if you do find markers, he wouldn't exclude them because as you increase population size, really what that does is it just gives you more power, but it doesn't really reduce the false positive rate.
Yeah, that makes sense. And another question, you were mentioning all the different models, like you had the MLM and then the compressed MLM, and then you showed farm CPU and then blank. How do you choose them? Do you recommend that people run all of the models and then use some sort of metric to choose the best one, or do you recommend to show the results from all of them? How do you do it?
What I personally like to do is I like to run kind of three generations of them. I like to run MLM, MLMM, and then B-Link, right? Because B-Link is going to give you the most power. It's going to run the fastest and can find multiple markers. However, sometimes I feel like B-Link gives me suspect results, or I feel like something went wrong in the algorithm, and I just get all these really rare alleles showing up as significant, and it just doesn't seem right. And then MLM-M is a little bit more conservative than B-Link, right? So it doesn't have as much kind of spurious markers that show up in the algorithm. It's a little bit easier to understand. And then MLM is even easier, but it has a really low power and low false positive rate. So I like to run the three, make sure they kind of get similar things. Um, I think at the end of the day, B-Link is probably the best one to run as far as power, and the false positive rate is pretty low. But it is nice to see how the older models do and compare it to what you find in the newer model, especially with that leading or the most significant um, S&P I like to see.
Cool, excellent. We have one more question here. Um, what about dioecious plants like Palmer amaranth? Can you use GWAS to identify, for example, differences in secondary sex traits in relation to, for example, response to abiotic stress?
Uh, so by secondary sex traits, do they mean kind of things that tag along with the sex of the plant, right? So let's say if it's male, maybe it's a little bit less abiotic stress tolerant or something.
Yeah, yeah, something like that. I would say like a correlation or like we call confounding of the traits.
So one thing you could do uh, is actually include the sex of the plant is a fixed effect covariate, right? And then when you include that in the model, if you're more interested in the secondary trait, like you already know the genes that explain the sex, but you want to know within each sex what genes are affecting the abiotic stress response, I would just fit a fixed effect covariate for sex, right? And then correct for it in the model. With the fixed effect models, and most of these GWAS models allow this, you can fit other fixed effect variables, like if they're grown in different environments or if they're of different sexes or anything you can think of that you might need to correct for, you can include as a fixed effect variable.
Cool. That makes sense. Um, and I don't think we have any other questions here. I do have one more, and it's more asking your opinion. Um, so at some point in your slides, you mentioned that we in weed science, we don't have SNP arrays or genotype chips. Do you think that's the reaction or maybe the next step that we should go in weed science to get develop SNP arrays for some species and then make them available to everybody who are interested in genotyping a specific species?
I actually think that it is not the direction we should go. And the reason I say that is because sequencing costs keep going down and SNP arrays have their own drawbacks. And so one thing that happened was in corn, when they came up with those GBS methods, they noticed all this diversity they didn't see before because when you make those SNP arrays, right, you only capture the diversity of the population you screen for those SNP arrays. So then if you get stuff from outside of it, it can look like the variation is really low, right? But it's just not capturing that variation. And so once they started using GBS and corn, they could see all this diversity they couldn't see before. And in crops, it made sense because the SNP arrays made it way cheaper. And if there were SNP arrays today, it would be much cheaper for weeds. But I think as sequencing costs keep going down, that just doing whole genome shotgun sequencing will eventually just become the cheapest way to do it. And then it'll be more of the computational methods and kind of haplotype phasing that people will think about, right? So if they could do shotgun sequencing and then phase their haplotypes super easily, that might be a cheaper and better solution in the next 5 or 10 years than actually coming up with array chips.
Yeah, that makes sense. Yeah. Cool. All right. Thanks so much, Samuel. I appreciate your time and good to see you again.
Yeah, thanks for having me.
Yeah. All right. Thanks, everyone. I will see you all next time in late November. We'll be sending out announcements pretty soon.
All right, thank you.
Bye.
Bye.